During the hydrolysis of sucrose, we can expect the product of alpha D glucose and beta D fructose. For a polypeptide hormone, we can give the example of insulin or with a glucagon. Denaturation of the protein is very important. Remember, for writing the definition with one example, you'll get a 3 marks. When you apply the electrical field, these are the amino acids which are containing both the positive charge and the negative charge, and it will move towards the respective electrodes. Hello my dear students, a warm welcome you all. Myself is Purnima, lecturer in Department of Chemistry at Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. My dear students, welcome back to the revision class of Unit 14, Biomolecule. Before going to discuss with this unit, let's see the weightage of this unit. From this unit, there is no 1 mark, 2 mark and a 3 mark question, but directly we can expect the 2 5 mark question and totally it carries 10 marks. Remember, if it is asked for 5 mark, there will be no direct question for the 5 marks at all. So it has been split into 3 plus 2 as well as with the 2 plus 2 plus 1. So that is why I have listed out the 1 mark, 2 mark and a 3 mark important question from this unit. Let's see what are the 1 mark question can also be raised in the 5 marks. Okay. So the first question will be goes like this. What are the expected product of hydrolysis of sucrose? During the hydrolysis of sucrose, we can expect the product of alpha D glucose and beta D fructose. Name an amino acid which are containing sulfur. So, we can expect the amino acids which are containing the sulfur also. The best example is cysteine as well as with the methanoin. So, these two are only the amino acids which consist of sulfur in their amino acids. Coming into the Zwitterion. It is a question can also be asked for 2 mark. Sometimes can also be asked for 1 mark. Okay. So, what is Zwitterion? These are the amino acid dipolar ions which are carrying both positive and negative charges, which moves either towards the cathode or either towards the anode in the electrical field. So, when you apply the electrical field, these are the amino acids which are containing both the positive charge and the negative charge, and it will move towards the respective electrodes. Okay, either it can move towards the anode, either it can move towards the cathode also. So, what is isoelectric point? The pH at which amino acids will be acting like a Zwitterion in the aqueous solutions are known as what? Isoelectric pH or the isoelectric point. Okay. So, at a particular point, the Zwitterion will be formed in an amino acids which consists of both the positive and the negative charge. Okay. Such point are said to be isoelectric point. Next question, what is polypeptide bond? It is a amide bond which has been formed between the 2 amino acids. If 2 amino acids are there, between the 2 amino acids, the bond which has been formed, that is said to be a polypeptide bond or peptide bond, we say generally. Next question, what is a nitrogenous base present only in the DNA, not in the RNA? We know that in a DNA, we have adenine, guanine, thymine and cytosine. Correct, no? So, present in case of DNA, we have a nitrogen base known as thymine which has been present only in the DNA, not in RNA. Like that, we also have a nitrogen base in RNA, not in DNA. That is nothing but a uracil. Okay. So, it is also known as AGTC here. AGUC. Okay. So, it is a thymine which is present only in the DNA, whereas in case of RNA, okay, we have only the uracil which is a nitrogenous base. Next, name an hormone which controls the carbohydrate metabolism. We know that in our body, the insulin is only the hormone which regulates the carbohydrate metabolism. If the carbohydrate content is too high, this insulin production which has also become too high, so that the patient may also become a sugar patient. Okay. Next, how many peptide bonds are present in pentapeptide? If you see that the peptide linkage, it always formed between the 2 amino acids. Okay. If 4 amino acids are there, it is a pentapeptide bond. So, which means to say that how many amino acids will be there? There are 5 amino acids will be there, and between these 5 amino acids, how many peptide linkage will be formed? You can see that there are 4 peptide bonds will be formed between 5 amino acids. Hence, it is known as pentapeptide. So, how many bonds will be formed? 4 bonds. Okay. Between the 5 amino acids, only 4 polypeptide bonds will be formed, or peptide bonds will be formed. Next question. Name the water insoluble component of the starch. So, if you see the starch, we have a water insoluble component also, and that is known as amylopectin. It is known as what? Amylopectin. So, these are the types of 1 more question which will be frequently asked in a part D, which carries 5 marks. Okay. Again, remember, in the 5 mark, there will be no direct question. It has been split into 2 plus 2 plus 1. So, that is why I have listed out the few important 1 mark question. Next 2 mark question we see now. Write the Harvard structure of the glucose. It is very, very important to know that the writing part of the Harvard structure of the glucose. If they don't ask you to write a Harvard structure, they can give you the important reactions which is exhibited by the glucose. Like in the structure elucidation of the glucose, how many bonds will be there in the glucose? For example, how the glucose will exhibit the 5 OH bonds, and what is the nature of that carbonyl group, whether it is a aldehyde group, whether it is a ketonic group which is present as the functional group, they can ask you the question. But most of the questions, it is mainly based upon the Harvard structure. So, be cautious with the structural elucidations of the glucose also. We can expect the 2 mark question. They can give you any condition to write the reactions of the glucose. How does it shows a 6 carbon chain, or with the presence of 5 OH bond, or with the presence of the functional group. Okay, keep that in mind. So, we will see with the Harvard structure of the glucose. We know that the glucose, which is a long chain carbon, which is having the 6 carbon atom. You can see that 1, 2, 3, 4, 5. And when we are writing this carbon, so 1, 2, 3, 4, 5 and 6. So, it is CH2OH, and this is CHO, and we have OH, H, OH, and so this is the structure of the glucose. Okay. This can also be written in the form of Harvard structure. You can see that this is the first carbon atom, 1, 2, 3, 4, 5 and 6 carbon atom, which is forming a bond with an oxygen. So, it is a alpha D glucopyranosis. Similarly, we can rewrite just by arranging the OH and H below and up, we can write the beta form. This is a beta D plus glucopyranosis. So, in the exam, not only they are going to ask with the writing the Harvard structure of the glucose, they can also go with the maltose, fructose as well as with the sucrose. So, all the 4 different types of the biomolecule, you must know that. So, one is glucose, fructose, maltose as well as with the sucrose. Okay. Next, we will say with the one more example. Write the Harvard structure of the maltose. So, the glucose and maltose, which is more frequently asked question. So, this is a Harvard structure of the maltoses, where you can see that the first and the 4th carbon atom, they have linked together by a glycosidic linkage. Okay, and it is known as alpha D glucose, and this is a alpha D, or you can say that beta D glucoses also. So, you can see that there are 5 carbon atom will be there, and this is the 6th one which are forming a bonding with an oxygen. So it is 1, 2, 3, 4, 5 and 6 carbon atom. Okay. So this is how we can write the structure of the maltose is. Remember, while writing, you should write only the 6-membered ring. Okay. Don't write with the 5-membered. Okay. It's a wrong representation. Next, we have what are the essential and non-essential amino acids? Give an example. So you need to write the definition with the essential and non-essential amino acids. We all know that essential amino acids are the amino acids which is required for the body, but they cannot be synthesized within our body, and it should be taken through the diet. Example, the valine and leucine. The best example is vitamin C. Okay, the vitamin C we take through the diet because it is not synthesized in our body. It should be taken through the diet. Like that, the amino acids, if it is not synthesized in our body, we taken through the diet, and it is very much essential for our body. Coming into the non-essential amino acid, even though it is synthesized in our body, but it's not required in an essential quantity. Example, the glycine and alanine. Okay, one example you must remember for this. Next question, write the differences between the fibrous and the globular protein. So we know that fibrous proteins, in the sense, these are the polypeptide chain run parallel and held together by a hydrogen bond and a disulfide bond, and they looks like a fibers. Okay. Coming into the globular protein, the structure will be results when the chains of polypeptides are coiled to form a spherical shape. Such proteins are generally insoluble in water and they are soluble in water. Example, keratin, the protein which is present in our hair, and insulin and albumin, these are the protein which is present in our blood. Next, 3-mark question. Give an example for each type of the hormone. One is polypeptide hormone, amino acid derivatives, and a steroid hormone. For all these things, which carries 1 mark. You need to give an example. For a polypeptide hormone, we can give the example of insulin or with a glucagon. For the amino acid derivatives, you can write any one of the amino acid derivatives because there are 20 amino acids will be there. So, 1, 2 example you can write like a thyroxine or with a epinephrine. Next, steroid hormone or example, a testosterone and a progesterone you can write because these are the hormone which regulates the 6 hormones in our body. Next question, write the functions of the following hormone. The first one is insulin, thyroxine and estrogen and androgen. So these 3 again will carries 1 mark. We know that insulin will maintain the blood sugar level, thyroxine will maintain the growth and development in our body, estrogen and androgen which are mainly helpful for the development of the secondary sexual characters. Okay. Next question, define the denaturation of a protein. Give an example. Denaturation of the protein is very important. Remember, for writing the definition with 1 example, you'll get a 3 marks. The loss of biological activity of the protein, when a protein is in a native form and it is subjected to the physical change like a change in temperature, changes in the chemical changes such as change in pH, etc. They are said to be denaturation of the protein. A best example is coagulation of the egg white on boiling or curding of the milk. So, when the protein, their physical and chemically, if you change their state, either in the form of temperature, either in the increasing the pressure, either, by altering the pH also, we are changing the nature of the protein, either in the physical, either in the chemical way. Such process we call it as a denaturation of the protein. Best example is curding of milk. Okay, where the physical state of the milk which has been converted into a liquid state, okay, and again chemically we are converting the milk into a curd. Similar way, the coagulation of the egg white is the best example upon boiling. Next, we have write the biological functions of the nucleic acids because it is a biomolecules. In the first part, you are going to study only about the biomolecules and its structure. So, in the second way, you are going to understand the structure of the nucleic acids. In the biomolecules, we have two parts. One is introduction to the carbohydrate. Next, we are going to understand more about the protein, then amino acids, then with the vitamins, and lastly with the nucleic acids. From any other parts, definitely one question will be asked for the 5 mark, okay? Either in the carbohydrate part, either in the vitamins part, either in the protein, either with the nucleic acid part. If it comes to the nucleic acid, we have much more important question is writing with the biological functions of the nucleic acids. Coming into their biological functions, we know that DNA is only the hereditary material which carries or transmit the characters from one generation to the next generations. Okay, DNA is a chemical basis for the hereditary and may be regarded as a reserves of genetic information. A DNA molecule is capable of self-duplication during the cell division, and identical DNA strands are transferred to the daughter cells, and this synthesizes a protein in the cell. Actually, the proteins are synthesized by various RNA molecule in a cell, but the message for the synthesis of the particular protein is present only in the DNA. These are the biological functions which are exhibited by the nucleic acids. Hope you all understood with this unit. Remember, in the biomolecule, there are 4 parts will be there. One is carbohydrate, proteins, vitamins, amino acids, and nucleic acids. From all other 4 parts, any questions can be asked for 5 mark. Be cautious with the writing. If they ask you to write for the carbohydrate, definitely next questions will be with the amino acids or with the nucleic acids. So, please think and write with the exams. I'll come back with the next unit. Thank you.